So welcome. In this session, we'll be looking at gluconeogenesis, which is the formation of glucose from non-carbohydrate precursors. Welcome all. My name is Wallace Munyiri, and I'm the redactor. So, gluconeogenesis. Some tissues in the body require glucose as a metabolic fuel. So it is imperative to supply these tissues with glucose. Okay. Now, we can do this by either using liver glycogen, okay, the glycogen stores in the liver, but these stores, they provide these tissues for with glucose for up to 24 hours. After 24 hours, those stores are depleted of glycogen. So we have to form we have to form glucose from non-carbohydrate precursors. Now, that process of formation of glucose from non-carbohydrate precursors is now what is known as gluconeogenesis. And it mainly takes place in the liver, okay, and the kidney, but the liver mainly, up to 90% in the liver. But during severe fasting, the kidneys usually become the major glucose producing organs, okay? Now, how does this gluconeogenesis occur? It cannot occur simply by reverse of the processes of glycolysis. No. Some steps in glycolysis are simply reversible, but others are irreversible. In fact, we have the 10 steps of aerobic glycolysis, okay? In those 10 steps, 7 are reversible. 3 of them are irreversible. Let me remind you the 3 of them which are irreversible. The reaction catalyzed by the hexokinase or glucokinase. Also, you have the other reaction which is catalyzed by the phosphofructokinase 1, and also the reaction which is catalyzed by the pyruvate kinase. Those 3, you can see that all of them are kinases. Those 3 are irreversible, okay, and they have to be bypassed. They are usually bypassed, and I will show you how we bypass them, okay? So here we are. You can see this reaction catalyzed by pyruvate kinase here. Also, this reaction here, label number 3, catalyzed by the phosphofructokinase 1, when you are moving from fructose 6-phosphate to fructose 1,6-bisphosphate. Also, this reaction when you are phosphorylating glucose at position number 6 to form glucose 6-phosphate catalyzed by the hexokinase. Those 3 reactions are irreversible, and they are bypassed by these 4 reactions, okay? So let's go now to see which substrates can you use to form glucose. Now, gluconeogenic precursors are molecules that can be used to produce a net synthesis of glucose, okay? Now, the most important gluconeogenic precursors are glycerol, okay? Let me take a pen. I see, I usually write with pen. We have glycerol, we have lactate, also alpha keto acids, and alpha keto acids are usually obtained by from the metabolism of gluconeogenic amino acids. Remember the 20 amino acids? All the 20 amino acids are gluconeogenic, apart from only 2, leucine and lysine, which are purely ketogenic, okay? But all the others are glucogenic. But we have others which are both glucogenic and ketogenic. When I say ketogenic, I mean they produce ketone bodies. When I say glucogenic, they can produce glucose. Now we have a category of amino acids which are both glucogenic and ketogenic, such as tyrosine, phenylalanine, tryptophan, isoleucine, such. They are both, but leucine and lysine are purely ketogenic. They can never produce glucose, okay? But the other 18, they can produce glucose, okay? Now, let's look at how glycerol can be used to produce glucose. It is usually produced by from the hydrolysis of triacyl glycerols, okay, in the adipose. Now, once you remove the fatty acids from the triacyl glycerol, you remain with the glycerol backbone, which is usually delivered from the adipose tissue to the liver by blood, and where it is phosphorylated by glycerol kinase This enzyme, glycerol kinase, is present only in the liver. Okay. Once phosphorylated at position number 3 to form glycerol 3 phosphate, it is further oxidized by glycerol 3 phosphate dehydrogenase to form dihydroxyacetone phosphate. Dihydroxyacetone phosphate is an intermediate of the glycolysis and gluconeogenesis. Okay, remember dihydroxyacetone phosphate and glyceraldehyde 3 phosphate are interchangeable. A reaction catalyzed by the triose mutase. Okay, now lactate. How can you get glucose from lactate? Now, lactate is obtained from the anaerobic glycolysis, where we convert pyruvate to lactate, a reaction catalyzed by lactate dehydrogenase. Now, lactate can be converted back to pyruvate by the same enzyme. Now, lactate from anaerobic glycolysis is usually released in the blood by exercising muscle, skeletal muscles, and also by the cells that act mitochondria, such as red blood cells. Okay, now this lactate is taken up in the Cori cycle and is usually taken up by the liver and oxidized to pyruvate that is then converted to glucose. Okay, then it is released back into the circulation, the Cori cycle. This is the Cori cycle. You can see here lactate is being produced by the exercising muscle during an anaerobic glycolysis. Then it is released into the blood and it is transported to the liver, where this lactate undergoes gluconeogenesis to form glucose, which is then released back to the blood and used up by the muscle again. Okay, this is the Cori cycle. And please don't confuse this Cori cycle with the Cahill cycle. Okay, the Cahill cycle is also known as the alanine pyruvate cycle. Please don't confuse the two. This is the lactate, glucose lactate cycle. The other one is glucose alanine. Sorry, not glucose pyruvate. Glucose alanine. Glucose alanine cycle is also known as the Cahill cycle, but this is glucose lactate cycle, also known as the Cori cycle. Now, how will the amino acids give us glucose? Amino acids are usually produced by the hydrolysis of tissue proteins that are the major sources of glucose during a prolonged fast. Now, their metabolism usually generate alpha keto acids. Examples are the pyruvate, which can be obtained from alanine, which can be then converted to glucose, or alpha ketoglutarate that can either enter that usually enter the TCA cycle to form oxaloacetate, which is a direct precursor of phosphoenolpyruvate. Okay, note that acetyl, please note this, acetyl-CoA and compounds that give rise only to acetyl-CoA, such as acetoacetate, leucine, and lysine, cannot give rise to glucose. Why? Because the reaction, which is catalyzed by the pyruvate dehydrogenase complex, the conversion of pyruvate to acetyl-CoA, that reaction is irreversible by nature. It is irreversible and it cannot be bypassed. Okay, so compounds that will give rise to acetyl-CoA, okay, such as leucine and lysine, the purely ketogenic amino acids, they cannot give rise to net synthesis of glucose. Okay, now the reactions involved in gluconeogenesis. Now, 7 glycolytic reactions are reversible, 7 of them, and are used in the synthesis of glucose from lactate or pyruvate. However, 3 glycolytic reactions, the one which are catalyzed by the kinases, but I will show you one kinase. I will show you one kinase which is reversible. That kinase, I don't know what is wrong with it, but it is a kinase, but its reaction is reversible. But the other kinases, the three other kinases, their reactions are irreversible, and they are usually circumvented or bypassed by 4 alternate reactions that are energetically, that usually that usually favor the synthesis of glucose. Okay, we are by circumventing those 3 reactions, we are favoring gluconeogenesis rather than glycolysis. So let me show you. Here you can see the pyruvate kinase. This reaction is reversible and it is bypassed by these two. Okay. But then here, okay, let me first show you the reversible ones. Then here the reaction characterized by the phosphofructokinase 1 here from this phosphofructokinase 1, it is irreversible and it is bypassed. Then from glucose here to glucose 6-phosphate, a reaction characterized by the hexokinase, it is also irreversible. So it has to be bypassed or circumvented. Okay. But here we have one kinase which is actually reversible, the phosphoglycerate kinase. This kinase is reversible. This reaction is reversible, despite this being a kinase. Okay, please remember that. But the other kinases, their reactions are irreversible and they have to be circumvented. Now, let's go back here and let's start here. Imagine you have pyruvate, okay? And pyruvate can be obtained from some amino acids, such as, I think alanine can give you pyruvate. Now, once you have pyruvate, you must convert this pyruvate to phosphoenolpyruvate and bypass this reaction catalyzed by the pyruvate kinase because it is irreversible. Okay, so pyruvate carboxylation. Now, the first roadblock to overcome in the synthesis of glucose from pyruvate is the irreversible conversion in glycolysis of phosphoenolpyruvate to pyruvate by pyruvate kinase. When converting phosphoenolpyruvate to pyruvate, this reaction is characterized by the pyruvate kinase, and this reaction is irreversible. So you have to bypass this reaction. How will you do that? First, you must convert this pyruvate to, you must convert it to oxaloacetate. You must, and the conversion of pyruvate to oxaloacetate is carboxylation. So a carboxylase will be used, the pyruvate carboxylase. Then you convert the oxaloacetate to phosphoenolpyruvate, a reaction catalyzed by the phosphoenolpyruvate carboxykinase. Okay, so those two will bypass this reaction. So first convert this pyruvate to oxaloacetate, then the oxaloacetate to phosphoenolpyruvate. Okay, now what if lactate was the precursor? Now this lactate is first converted back to pyruvate by the lactate dehydrogenase. Then the pyruvate is transported into the mitochondria and it will be carboxylated by pyruvate carboxylase, okay, to oxaloacetate. Then this oxaloacetate is converted to phosphoenolpyruvate directly by the mitochondrial isozyme of phosphoenolpyruvate carboxykinase. Okay, then the phosphoenolpyruvate is transported to the cytosol where it will continue with the gluconeogenesis. Now, oxaloacetate, let me now, I think here we are, let's start here. Here you have pyruvate, okay, pyruvate here. This pyruvate must undergo carboxylation. You add a carboxyl group at this position. Yeah, that is where you have to add a carboxyl group. And this reaction is catalyzed by the pyruvate carboxylase. Now, pyruvate carboxylase, shown here, this is a pyruvate carboxylase. Okay, it has a lysine residue, and to the lysine residue, biotin is attached there. So biotin is the coenzyme here. And this biotin, it acts as the carrier of the carboxyl group. Okay, and you can see in this reaction where we are somehow activating the enzyme by adding carbon dioxide in form of a bicarbonate ion. This reaction, you can see it requires ATP. Okay, and it is activated when we have high amounts of acetyl CoA. Okay, so once we like load the enzyme with the carboxyl group here, this carboxyl group is one which is transferred to this position on pyruvate to form your oxaloacetate. Okay, and the enzyme is now goes back to its original form. You can see the carboxyl group has been removed here. Okay, then the oxaloacetate is converted to myrate. Why convert oxaloacetate to myrate first? Because we don't have a transporter for oxaloacetate. You first must convert the oxaloacetate to myrate by the mitochondrial myrate dehydrogenase. Then once you transport it to the cytosol, this is the mitochondria matrix, then this is the cytosol. Once in the cytosol, you convert it back to oxaloacetate by the cytosolic myrate dehydrogenase. And you can see in this reaction, we are producing NADH. This NADH has some uses, and I will show you. Please keep this in mind. Okay. Then oxaloacetate is converted directly to phosphoenolpyruvate, a reaction catalyzed by the cytosolic phosphoenolpyruvate carboxykinase. Okay. And you can see we are using GTP, not ATP. We are using GTP. And we are also producing CO2. And this CO2 is the one which can diffuse through the mitochondria and form this bicarbonate ion, which will be used in the carboxylation of pyruvate to oxaloacetate. Okay, so here the process I've just described is if pyruvate was the precursor. What about if lactate was the precursor? So you will start here with the lactate here in the let's call it lactate in the cytosol. This lactate will be first converted to pyruvate. Let's call it pyruvate, PRY, pyruvate by the lactate dehydrogenase. Then this pyruvate is usually transported to the mitochondria and we come here. Then it will undergo carboxylation by the pyruvate carboxylase to form oxaloacetate. Then something interesting will occur. Instead of it being transported out of the mitochondria to the cytosol, it is converted directly to phosphoenolpyruvate by the mitochondrial phosphoenolpyruvate carboxykinase. So this is the mitochondrial isozyme of phosphoenolpyruvate carboxykinase. But if pyruvate was the precursor, it has to be transported back to the cytosol so that the oxaloacetate can be carboxylated. In fact, it is decarboxylated by the phosphoenolpyruvate carboxykinase. Okay, so please note that there is a difference. If lactate was the precursor, the oxaloacetate will be converted to phosphoenolpyruvate by the mitochondrial isozyme of phosphoenolpyruvate carboxykinase. But if pyruvate was the precursor, it will be converted to the oxaloacetate will be converted to phosphoenolpyruvate by the cytosolic isozyme of phosphoenolpyruvate carboxykinase. Please note that. Now, we have formed the phosphoenolpyruvate, and here it is. Now, here I hope you have done glycolysis, so you know these are the reversible reactions. So phosphoenolpyruvate here will be converted to 2-phosphoglycerate by the enolase. Remember one interesting thing about enolase, it is that it is inhibited by fluorine, and that is why, and you ask yourself, why is fluorine used in toothpaste? Then 2-phosphoglycerate is converted to 3-phosphoglycerate by the phosphoglycerate mutase. Then 3-phosphoglycerate mutase is converted to 1,3-bisphosphoglycerate by the phosphoglycerate kinase. This is the kinase which is the reversible one. Then 1,3-bisphosphoglycerate is converted to glyceraldehyde 3-phosphate by the glyceraldehyde 3-phosphate dehydrogenase. Now, this reaction of converting 1,3-bisphosphoglycerate back to glyceraldehyde 3-phosphate requires NADH and the hydrogen ion. Where did you produce the NADH and the hydrogen ions? You produced them when you are converting myrate to oxaloacetate, a reaction catalyzed by the cytosolic myrate dehydrogenase. Remember the reaction I told you to remember, the NADH and the hydrogen ion? That is where they usually come and are used in this reaction. Then once we form glyceraldehyde 3-phosphate, two of them, one of them is usually converted to dihydroxyacetone phosphate, a reaction catalyzed by the triose mutase. Remember, dihydroxyacetone phosphate, this is the entry point of glycerol. Remember, I told you glycerol first is phosphorylated by the glycerol kinase to form a glycerol 3-phosphate. Then glycerol 3-phosphate undergoes oxidation by the reaction catalyzed by the glycerol 3-phosphate dehydrogenase to form dihydroxyacetone phosphate. Now, dihydroxyacetone phosphate and the glycerol 3-phosphate combine to form fructose 1,6-bisphosphate, a reaction catalyzed by aldolase A. Okay. So let's continue. Now you have formed the fructose 1,6-bisphosphate. This reaction, now let me take back the pen. The reaction of converting fructose 6-phosphate to fructose 1,6-bisphosphate was being catalyzed by the phosphofructokinase 1, and it is irreversible. So we circumvent it using this reaction, which is catalyzed by the fructose 1,6-bisphosphatase. Okay, and you can see the bisphosphatase, we are removing the phosphate group at carbon number 1. This one is the one which you are removing. It is removed. And this reaction is an important regulation site for gluconeogenesis, because fructose 1,6-bisphosphatase is inhibited by AMP. Also, it is inhibited by fructose 2,6-bisphosphate. Now, AMP shows the energy levels in the cell. If we have high AMP levels, it means the ATP levels are very low. So if ATP levels are very low, you want glycolysis to take place so as to produce more ATP. So you don't want gluconeogenesis to take place and you don't have energy. So that is why AMP inhibits that. Also, fructose 2,6-bisphosphate depends on insulin glucagon levels, okay? And it is usually produced when the glucagon levels are high. Let me show you. And when insulin levels are low, let me show you how you produce fructose 2,6-bisphosphate here. When glucagon levels are high, and remember they act via the G protein coupled receptors, and they activate adenylyl cyclase to produce cyclic AMP. Now, the cyclic AMP will activate a protein kinase A, and the protein kinase A will phosphorylate will phosphorylate this. We have a bifunctional enzyme, and the protein kinase A will phosphorylate the phosphofructokinase 2 domain. Okay. Once we phosphorylate that domain, the fructose 2,6-bisphosphatase will be activated so that we can convert now the fructose 2,6-bisphosphate to fructose 6-phosphate, which will now enter the gluconeogenesis. Okay. And once you remove this fructose 2,6-bisphosphate, you can see this the inhibition is removed. Okay. So glucagon will promote gluconeogenesis, but insulin will inhibit gluconeogenesis. So that is that. Now, we have now formed fructose. We have now formed fructose 1,6-bisphosphate. Now this fructose 1,6 bisphosphate, I said it's converted to fructose 6-phosphate by the fructose 1,6 bisphosphatase. Now this fructose 6-phosphate is converted to glucose 6-phosphate. This reaction is reversible. So this is reversible, and it's usually characterized by the phosphoglucoisomerase. Okay, I think it is the glucose 6-phosphate phosphoglucose isomerase. Okay, and isomerase is reversible. Then we have now formed glucose 6-phosphate. We need to carry out dephosphorylation of this so as to get glucose. Okay, you need to remove the phosphate group at position number 6. Okay, now glucose 6-phosphate hydrolysis to it is usually hydrolyzed by the glucose 6-phosphatase to bypass the irreversible hexokinase or glucokinase reaction and provides an energetically favorable pathway for the formation of free glucose. Now, the liver is the primary organ that produces free glucose from glucose 6-phosphate, and this process requires a complex of 2 proteins. We require 2 proteins because glucose 6-phosphate is found in the lumen of endoplasmic reticulum. So you require first a glucose 6-phosphate translocase so as to transport the glucose 6-phosphate this from the cytosol to the lumen of endoplasmic reticulum. Then now you require the enzyme itself, the glucose 6-phosphatase, now to dephosphorylate a glucose 6-phosphate to form free glucose. Okay. Now you can have deficiencies of this enzyme. Like if you have the deficiency of glucose 6-phosphate translocase, you'd have type 1 glycogen storage disease, von Gieck, and it will be 1B, von Gieck 1B. But if you have the deficiency in glucose 6-phosphatase itself, you'd have von Gieck 1A. Okay, 1A. So you have type 1 GSD, as it is also known as von Gieck. It has 1A and 1B. 1A is if you have deficiency in glucose 6-phosphatase itself. 1B is if you have a dysfunctional glucose 6-phosphate translocase. Okay. Now, and this is the reaction from glucose 6-phosphate to D-glucose. Now, how many ATPs are used in this process? Now, remember that you must start with the 2 pyruvates. Okay, 2 pyruvates are converted to 2 oxaloacetates, and remember the reaction here was being catalyzed by the pyruvate carboxylase, and we required an ATP for each. So it is 2 ATPs are used. Then 2 oxaloacetate, for them to be converted to 2 phosphoenopyruvates, a reaction catalyzed by the phosphoenopyruvate carboxykinase, you require 2 GTP. But remember, GTP and ATP are interchangeable. Okay. Then the reaction of converting 3 phosphoglycerate to 1,3 bisphosphoglycerate, the reaction which is catalyzed by the phosphoglycerate kinase, and it was the reversible one, you also require 2 ATPs. Okay. So for each reaction, remember these two I've put here in bold. I mean two molecules of that substance. So you require 2 molecules of 3 phosphoglycerate to form 2 molecules of 1,3 bisphosphoglycerate. So and 2 ATPs will be required. So a total of 2 ATP plus 2 ATP, 4 ATPs, and 2 GTP, which are equivalent to 2 ATP. So you require an equivalent of 6 ATPs to form glucose from pyruvate. So you can see it is a very expensive process. So I think we have come to the end, and let me leave you with one quote. The man who asks a question is a fool for a minute, but the man who doesn't ask is a fool for life. That's all. Thank you for your attention, and please subscribe for more anatomy and biochemistry content. Thank you.